Enzymes are important to the body because they are necessary for many of the chemical reactions that occur within it. My name is Alyssa, and this video will help you understand how these enzymes are regulated so that they're not always functioning, especially when we don't need them to. We'll start by looking at the process of feedback regulation, and then investigate specifically how these enzymes are prevented from working. We'll then finish by discussing the chemical methods through which these enzymes are regulated. After watching this video, you will be able to recall the different kinds of enzymes and how their function is controlled within our bodies. You will be able to answer questions that ask you to think about the different kinds of inhibition and recall if and when these processes can be overcome. Feedback regulation occurs when the products of a metabolic pathway affect the activity of enzymes within that pathway. For example, this pathway proceeds from protein A to protein D by 3 chemical reactions. These reactions are facilitated by enzymes 1, 2, and 3. If protein D then controls the activity of enzyme 1, it is considered feedback regulation. This regulation can be negative, creating a negative feedback loop in which the presence of product D prevents enzyme 1 from functioning. This process helps maintain homeostasis once enough product has been made. This is the most common feedback loop because once enough of the product is made, that product can then limit its own production. Occurring much less common, the feedback loop can also be positive, where the products will bind to the enzyme and encourage it to work more, creating more product D. How do these products control how the enzyme functions? This has to do with the way in which the product binds to the enzyme and what effect it has on the active site. We'll start by exploring reversible inhibition, when a molecule can bind to an enzyme, cause an effect, and leave the enzyme without destroying it. Competitive inhibition is the first of 4 types of reversible inhibition. Without inhibition, a substrate and an enzyme will come together and a reaction will occur in the active site of the enzyme. In this case, a substrate is reacting in the active site and breaking down into 2 separate molecules. With competitive inhibition, the inhibitor competes with the substrate to bind to the active site of the enzyme. However, the inhibitor is inert and will not react there. It just keeps the space occupied so that nothing else can react. When this happens, the substrate can no longer enter the active site and reactivity doesn't occur. Because the inhibitors are competing with the substrate for the active site, this kind of inhibition can be overcome by increasing the concentration of the substrate so that it floods the active site more than the inhibitor does. We can visualize this kind of inhibition using a Lineweaver-Burke plot, which plots the inverse of the concentration of the substrate on the x-axis and the inverse of the rate of reaction on the y-axis. Remember that for these graphs, the maximum rate at which the enzyme can function is found to be the inverse of the y-intercept, and the Michaelis-Menten value is the negative inverse of the x-intercept. This graph shows that as the concentration of the inhibitor increases, the maximum rate the enzyme can function remains the same, because when the concentration of the substrate is a lot higher than the inhibitor, it will always outcompete the inhibitor and reaction can occur at its regular rate. It is only the Km values that differ in this kind of inhibition because the substrate concentration has to be higher to reach half the maximum velocity in the presence of inhibitor. Non-competitive inhibition occurs when the inhibitor binds to an allosteric site rather than the active site. In this case, the enzyme has both an active site and an allosteric binding site. Without inhibition, the substrate binds to the active site and breaks down. Note that the allosteric site remains available for binding. When the inhibitor binds to the non-catalytic allosteric site, it causes a conformational change in the active site that prevents the substrate from binding. No amount of substrate will overcome this type of inhibition because the active site is no longer in the proper conformation for binding or reactivity. A non-competitive inhibitor has equal affinity to the allosteric site of the enzyme as it does to the enzyme-substrate complex. This means that the inhibitor can bind to the enzyme when the substrate is already bound to the active site. This will effectively kick the substrate out of the active site and prevent the reaction from occurring. Using a Lineweaver-Burke plot again, we can see that increasing the concentration of the non-competitive inhibitor decreases the measured Vmax because less enzyme is available for reaction, but it does not affect the Km value because the active enzymes still have the same affinity for the substrate. Uncompetitive inhibition occurs when the inhibitor will only bind to the enzyme-substrate complex and not the enzyme alone. The enzyme still has both an active site and an allosteric site, but what happens is that the substrate will enter the active site as if it's going to react. Then, the inhibitor binds to the allosteric site and prevents the substrate from being released. This binding can only happen once the substrate has entered the active site due to structural changes in the enzyme that open up the allosteric site after the substrate has entered the active site. Look at the Lineweaver-Burke plot. We can see that increasing the inhibitor concentration lowers both Vmax and Km, producing parallel lines on the plot. Mixed inhibition is very similar to non-competitive inhibition, but occurs when the inhibitor does not have an equal affinity to both the enzyme and enzyme substrate complex. The inhibitor can either bind preferentially to the enzyme substrate complex, in which case binding of the inhibitor causes the substrate to be released from the enzyme, or the inhibitor can bind preferentially with the enzyme, preventing the substrate from entering the active site initially. Either way, depending on the inhibitor and enzyme combination, binding affinity will be stronger for one complex over the other, and the substrate will not react in the active site. The Lineweaver-Burke plot is then a mixture of non-competitive and uncompetitive plots. If the inhibitor binds preferentially to the enzyme, then it will increase Km. But if the inhibitor binds preferentially to the enzyme substrate complex, then it will decrease Km. Therefore, the intersections of these Lineweaver-Burke plots will not lie on either axis. Irreversible inhibition occurs when an inhibitor causes a permanent change in the enzyme that prevents it from functioning. The only way to increase the rate of reaction is to produce more enzymes. A common example of an irreversible inhibitor is aspirin. This drug binds to cyclooxygenase 1 to prevent it from making prostaglandins, modulators of pain and inflammatory response. More cyclooxygenase 1 must be synthesized if this process is to occur again in the future. Allosteric enzymes have allosteric sites, which are locations where other proteins or molecules can bind to regulate the activity of the active site. In this case, the molecule binding to the allosteric site is an inhibitor, which decreases the binding affinity of the substrate to the enzyme. Three of the kinds of inhibition that we have already covered are examples of enzymes used in allosteric regulation. There are, however, examples of allosteric activators, which will increase the binding affinity of the active site and the substrate. Enzymes can also be regulated by covalent modifications, such as phosphorylation or glycosylation. Covalently attaching or detaching a phosphate group can affect the activity of an enzyme. Whether these actions activate or deactivate the enzyme depends on the specific reaction in question. In glycosylation, a sugar molecule is covalently bonded to an enzyme to tag it for transport within the cell or to modify the activity of its active site. Many of the enzymes in the body would have disastrous effects if they were not tightly regulated. Some exist primarily as zymogens, or inactive enzymes. These enzymes only become active once another reaction occurs to remove the regulatory domain, or the portion preventing reactivity. An example of this is trypsinogen, a zymogen for the enzyme trypsin, which is responsible for digestion within the stomach. Once in the stomach, enteropeptidase causes a reaction that transforms trypsinogen to trypsin and opens up an active site. So that you can recognize these molecules on test day, remember that zymogens are frequently named with the suffix -ogen and are the inactive form of the enzyme. Let's test our skills with these practice problems on enzyme regulation. This question asks you to think about the Lineweaver-Burke plots for enzyme inhibition and about what values are affected during each type of inhibition. The answer is D. In uncompetitive inhibition, increasing the inhibitor concentration lowers both Vmax and Km. Vmax lowers because there is less enzyme available for reaction and the rate decreases, and Km decreases because the substrate concentration has to be higher to reach half of the maximum velocity of the reaction when in the presence of the inhibitor. This produces a graph that will have parallel lines. This final question is asking you to think about which enzyme regulation example is affected when there is an excess of substrate. The answer is C. Competitive inhibition occurs if the enzyme and inhibitor have the same affinity for a binding site. With equal concentrations of inhibitor and substrate, the rate of the substrate conversion is decreased because the chances are the same that the inhibitor or the substrate will be binding to the active site. Remember that if you increase the concentration of the substrate, it will flood the active site and the rate of the reaction will not decrease. Great job if you got those questions correct. In this video, we examined the different methods of enzyme regulation. We started by defining feedback mechanisms, in which the products of the reaction affect the activity of the enzyme required for reactivity. We also investigated the 4 types of reversible inhibition, where the enzyme is out of commission only as long as the inhibitor is bound to it, and this is only temporary. There is also irreversible inhibition, when the enzyme is permanently destroyed. A new enzyme must be made when there is a reaction that requires that enzyme to occur. Lastly, we touched on the three ways in which enzymes are regulated: through allosteric sites, covalent bonds to molecules, or by being released as zymogens, inactive forms of the enzyme. Thanks for watching this video, and good luck studying for your MCAT.